Now this is the growth curve of animal cells. On x-axis is weeks in a culture, and on y-axis is number of cells or passage number. Okay. Now see, this graph is like a sigmoidal curve. Okay. Now why here is weeks in a culture on x-axis is because animal cells require more time than microbial cells to grow. As we can grow microbial cells within 24 or 48 hours, animal cells need weeks for the growth. Okay, so this is say first same like a lag phase where primary culture is developed. Primary culture, the culture which is developed from using explant. Okay, then once the primary cell culture is grown, see here, after 2 weeks the first subculture occurs. First subculture is made, that is the cells from the primary culture, they are transferred from old flask to the new one or to the fresh medium. Okay, then the second subculture, third subculture, and so on after specific time interval. Okay, now after few subcultures, see here you can see the cells are growing exponentially, and at this point the cells are become transformed. Cells become transform in the sense they do not require any special growth factor or nutrient for their growth. No special requirements for their growth. They just grow indefinitely. Even if you don't add any growth factor into the medium, they can grow continuously. The cells are known as transform. Here at this stage the cell characteristics changes, and from this stage onwards the cell culture is known as continuous cell line. Okay, continuous cell line from the first subculture till the culture is transformed. This period, in this period, cells are known as cell cultures are known as cell lines, because here all after each subculture the cell culture becomes homogeneous. Homogeneity is maintained, that is only one or two types of cells are grown in a single flask. Okay, and few cells are transformed, they grow continuously, they form continuous cell lines, and then few cell cultures they will show senescence or they will die, since they have replicated for specific number of generations. Animal cells, they replicate 20 generations. Okay, for 20 generations. After 20 generations they show death. Even if you subculture, even if you provide them fresh nutrients, those cells won't grow, they die. Okay. Now the terms to remember: primary explant culture. It is a fragment of tissue placed at a glass or a plastic liquid interface where after attachment migration is promoted in the plane of the solid substrate. Tissue is placed on a piece of glass, and that glass piece is immersed into the liquid medium. Glass piece and the few factors present in a liquid medium promotes attachment of a piece to the glass or say to the surface, and after attachment the cells which grow, they migrate and form a layer of a culture onto that glass piece or plastic piece. Then the cell culture. Cell culture implies that the tissue or outgrowth from the primary explant is dispersed. Okay, from the primary explant culture the cells are dispersed mechanically or enzymatically into the cell suspension, which may then be cultured as adherent monolayer on a solid substrate or in a suspension culture medium. Okay, when you transfer cells from primary explant to the next medium, the term is used for used cell culture. Then the passage. Passage in normal terms is subculturing. Okay. In animal cell culture, subculturing is termed as passage because cell proliferation is often found in cultures. The propagation of cell lines become feasible. A monolayer or a cell suspension with a significant growth fraction may be dispersed by enzymatic treatment or simple dilution and reseeded or subcultured into the fresh vessel. This is known as passage. Transferring cells after formation of monolayer into the fresh medium is passage. So as this passage procedure is not as simple as we transfer microbiological cultures into the medium. This needs disaggregation of cells. Now since these are animal cells, they attach themselves to the surface and then grow. Okay, so this attachment is very firm. You have to remove this attachment so as to remove the cells from the substratum. Substratum is surface to which the cells are attached. You have to use enzymes for that. Those enzymes, they digest the cell adhesion molecules and the proteins which help cell to adhere to the substratum. Okay, these enzymes, they digest cell adhesion molecules and thereby release the cells into the medium. The cells come up into the liquid medium. Those cells are then observed under the microscope. Their vitality ratio, vitality is calculated. How many cells are live, how many are dead. The percent is calculated and then the cells are reseeded, that is re-inoculated into the fresh vessel. Passaging. Okay, then the cell lines. Daughter cultures formed after subculturing or passaging is known as cell lines. Daughter cultures formed after subculturing or passaging is known as cell line. Cell strain. When the cells are selected from a culture by cloning or by some other method, the subline is known as cell strain. When the cells are selected, picked up by cloning method or by any other method, the subline is known as cell strain, where you will find only one type of cell or 2 to 3 types of cells in a culture, known as cell strain. Monolayer culture. Monolayer culture, given the opportunity, animal cells attached to the substrate and normally the cells will be propagated in this mode. Now this monolayer formed in such a way that each cell is surrounded or is in contact with its neighboring cell. Okay, and this is how the layer of the cells is formed, known as monolayer. These animal cells, they do not stack one above the other in a culture. They form a monolayer which indicates these cells now have to be subcultured or passaged. Okay, the monolayer culture, the cells growing in a monolayer culture, they follow contact inhibition mechanism. Contact inhibition, when the one cell, say central cell, is in contact with the neighboring cell, the central cell stops growing, known as contact inhibition. Okay, then the anchorage dependence. Anchorage dependence, all animal cells, they are anchorage dependent. They need surface to attach themselves for their growth and proliferation. Suspension culture, derived from the cells that can survive and proliferate without attachment, that is, they are anchorage independent. These cells are generally cancerous cell or transformed cells. They grow in suspension cultures. Histotypic culture, these are high density cell cultures or a tissue like. Histotypic, histo is tissue like cultures excuse me, of one cell type. Then the organotypic culture, the presence of more than one cell type interacting as they might in the organ of origin, as they may interact in vivo. It is 3D culture of undisaggregated tissue retaining some or all of the histological features of the tissue in vivo. Cell culture we have already discussed. A culture derived from dispersed cells taken from original tissue, from a primary culture, or from a cell line or cell strain by enzymatic, mechanical or chemical disaggregation. Histotypic culture, this also we have discussed. Example we'll see: by cultivation of cells at higher densities in a filter well or a perfusion, and overgrowth of a monolayer in a flask or a dish, reaggregation in suspensions over agar or in a real or stimulated zero gravity, or using matrix such as collagen gel. This also we have discussed. Now we'll move towards primary culture. The cells, when surgically or enzymatically removed from organism and placed in a suitable culture environment, artificial environment of course, will attach and grow, are called primary cultures. The cells directly removed from the animal or say an individual cultured in artificial environment known as primary culture.